I was so hungry, I even ate the bulbs of tulips in October 1944.
Famine cannot affect the genes, but we do think that there might be effects of exposure to famine on whether genes are switched on or off.
One could go so far as to say that you can greatly influence what genes are switched on and off through diet and enzyme activity.
Welche Gene man anschalten kann oder abschalten kann.
The insights we've gained from models so far show clearly that the environment has an impact on our genes that can be passed on to subsequent generations.
Weitergegeben werden kann, auch auf die nächsten Generationen.
Everyone knows food and drink is good for the mind and body, but how much they affect us, our appearance and behavior, is not clear to most of us. Food doesn't just fill us up, it affects our genes. This relatively new science is called epigenetics, which means in addition to genetics, and looks into how all of this works. Annabel and Gemma Molero from Barcelona are identical twins. They have exactly the same genes. That's why they are particularly suitable to find out how humans change throughout their lives by virtue of their habits. At age 1, they still looked almost the same.
When we were little, we lived with our parents, of course. After university, we moved to England together. I lived there for a year. My sister moved to Mexico after 6 months. I lived in Mexico for 4 months. After that, we were properly separated. Before that, we'd always been together. That was when we started growing apart, in our habits, too.
Different homes also led to different lifestyles. Today they are easy to tell apart. But why? Their genes, responsible for their appearance, stay the same throughout life. Their eating habits give a first clue.
I shop so I can prepare something quickly. My diet isn't the best. I eat almost anything, except for some things I don't like, and a few things I'm allergic to. I eat a lot of pasta and meat. I do a lot of sports and try to eat a good diet. I eat a lot of fruit and vegetables. I think I have a balanced diet. I also enjoy cooking.
It is clear that the quality and quantity of food shape the body. But how permanent is the impact on the person as a whole, on the genes? Dr. Manuel Estella works in Madrid's Cancer Research Institute. He has a theory about the cause of the changes. He takes saliva samples which contain the young women's DNA. Their genes are still identical, but something has changed.
Fingerprint in der Gitarre? Isabel sus hermanos o no?
Pues sí. The nucleus contains chromosomes. They are made up of long DNA chains that contain all the hereditary information, the genes. The DNA is wrapped around chunks of protein called nucleosomes. When methyl groups dock onto the DNA, the nucleosomes draw close together. These methylated DNA segments can't be read anymore. The genes on them are switched off.
Here we did like a double-blind study. In one side, they took a questionnaire, different questions about lifestyle, what type of food they eat, if they were smokers, non-smokers. At the same time, we had the DNA data. we have all these DNA methylation status, and we mixed both data, and surprisingly, or not, we found that they had DNA methylation difference according to a different lifestyle.
Switching genes on and off could explain why identical twins start being different as soon as one of them makes a lifestyle change. But what does this DNA methylation do? What happens when genes are switched off? Esteye examined hundreds of twins in search of answers.
One of the most surprising cases were a couple of brothers, of twins, that they had very different lives, with different diseases, one and living in a big crowded city and the other in a small town without any disease. And if you look at the epigenetics, they were like different people, like they were not twins anymore. From epigenetics, although the genetics was the same. And this was one of the extreme cases that we found.
To scientists, the epigenome is a kind of circuit diagram that shows which genes are active and which ones aren't. He has been comparing these diagrams in his experiments with twins.
Here we see one study about twins, and these are the example of two twins that have the same disease. And you see a straight line, indicating that there are no epigenetic difference. These twins, they have a different disease, and you see all these points that appear here, outside the straight line, that indicate epigenetic alterations in these twins. These twins, with a different disease, with the right treatment, all the wrong points appear again in the straight line, indicating that epigenetic can be changed with the right drugs.
What causes epigenetic changes? What makes our genes go silent or suddenly be switched on? The main cause may be the food with which we deliver the necessary building blocks our body needs, or else drinks, or poisons like alcohol. Even the synthetic material we touch or whether we smoke plays an important role. The sun's ultraviolet rays and our stress hormones have an epigenetic influence. It is basically all the many influences of our lifestyle that switch our genes on and off and make us what we are. The crucial role of food in all of this is currently being researched in Amsterdam in the Netherlands through one dark chapter in the city's history. In the winter of 1944-45, the German occupation forces blocked the food supply of the Dutch as punishment. Suddenly, there was almost no food. Thousands died. The blockade lasted for almost 6 months. Children and pregnant women were particularly affected. Dr. Tessa Roseboom is examining the women who were pregnant at the time. She wants to research the long-term effects of their malnutrition. Lois Hose is one of them. When the jolly 87-year-old serves up meals today, it's hard to imagine what it was like for her waiting for hours and hours for food when she was pregnant, until she talks about the birth.
Before my delivery, they laid me on wet sheets for 3 days because of some kidney poisoning. They tried to induce my labor so my child would be born earlier. I had strong contractions and they had to tie me down because I was smashing everything. For a few days I was blind and couldn't talk anymore. My tongue hung here. That's all I can remember. It was terrible. He looked like a skinned rabbit, very thin and long. I came home with him but couldn't feed him because I didn't have anything. I couldn't go out with him because I didn't have a coat or pram. The cold was extreme in 1945. How was I supposed to care for a child? Nobody helped me.
En hoe moet je dan met je kindje? Niemand die je helpt. Dat is ze behouden
It's
geweest.
terrible to have to give your own child away.
Maar het is erg als je je kindje weg wil brengen.
These are the kinds of destinies piling up in the files in her office. House calls and months of going through the documents give her the chance to create a unique study. Nowhere else can the consequences of extreme malnutrition of pregnant women and unborn babies be followed for such a long period of time as during this 6-month food blockade. Hundreds of women from different walks of life and their children were all equally affected. They all had to deliver their babies under the same conditions in Wilhelmina Hospital.
We have the birth records of babies born in Amsterdam in this Wilhelmina Gasthuis around the time of the famine, so before, during and after the famine. And we have followed them up until adulthood, and we're studying their health, comparing the ones who were exposed to famine in pregnancy and the ones who were born before or conceived after the famine.
Roseboom found several generations in the Fafiani family. For hours, the scientist has questioned the mother and daughter about their winter of famine and the following decades.
I was very worried about my child. I thought she had to grow. But how? I wasn't able to buy milk. That's important for a child. I could only give her the breast, but I didn't have enough. It was awful.
Today it is slowly becoming clear that the privations of the past have not remained without consequence for the daughter.
I was around 17 when I got kidney stones. Then I got high blood pressure, and my cholesterol level was raised. No, first I got breast cancer, then my cholesterol went up. I've dealt with a lot in my life.
The fact that her health has been this bad has made her think.
I don't have an explanation for it. It must have been that winter of famine, because my sister is a few years younger and doesn't have any complaints. Neither does my brother.
Want mijn zus is een paar jaar jonger als ik en die heeft daar allemaal geen problemen mee.
I have all of the problems.
Ik ben degene dus die alle probleempjes heeft.
Clearly, something in the emaciated baby bodies saved some information about how to deal with food for the rest of their lives.
The interesting observation was that people that are born with low birth weight, they're born small, they have a very higher risk of developing diseases later on in life, like type 2 diabetes and cardiovascular disease. And so this is called developmental programming of adult disease.
A famine, no matter how bad it is, can't change our DNA. But still, malnutrition has permanently switched genes on or off in the children of the post-war generation. A strange reaction by nature whose effects are still apparent today.
What is fascinating from the epigenetics point of view is that this is something that happens earlier on, but only as a biological effect much later on. And so because epigenetics confers cellular memory and are very stable marks, they can propagate the memory in early life to many decades later.
Just how dramatic the effects of genes being switched off by food can be seen in animal studies.
Nun, die Fellfarbe dieser beiden komplett genetisch identischen Tiere, also Zwillinge.
These animals are genetically identical. But I can change the color of their fur if I add more soy products to their diets, for example.
Hinzugebe. Und diese Hormone, die in diesem
The
Soja drin
hormones in
sind,
soy
bewirken,
cause the gene responsible for fur color to be changed.
Hautfarbe, also hier Fellfarbe natürlich verantwortlich ist, verändert wird.
But can these results found in mice simply be transferred to humans? Do the foods we consume every day affect our genes just as much? One clue could come from a southern Indian ethnic group who don't eat any animal products because of their religious convictions. Adults and children generally enjoy the best of health despite inadequate hygiene by Western standards. Their diets seem to give them everything they need. Some of these families emigrated to Europe and continued eating the way they did back home. But many of them fell ill quite quickly. The reason? The meticulously cleaned vegetables no longer contained the bacteria that supplied the vegans in India with the epigenetically important vitamin B12. In Japan, another epigenetic phenomenon was discovered, which became known as the Japanese paradox. Despite being an industrialized, hectic society, the country has a lower incidence of cancer than other comparable countries. The solution to this riddle was green tea. This drink is so widespread in Japan that it even suppresses the cancer statistics. If the unfermented tea leaves are boiled up with water, a unique chemical becomes available. This substance reacts to a certain gene that often becomes inactive with age. Green tea removes the methyl groups from the switched-off genes and simply switches them on again. The activated gene starts producing the body's own substances again, which fights cancer.
I think that's exactly the excitement, is that in theory, every single disease could have a very important epigenetic component, because virtually all genes in the genome are regulated by epigenetic marks.
Are epigenetic markers, genetic switches, also responsible for mental illnesses? New research suggests that the causes of schizophrenia, which are as yet uncertain, must be epigenetic in nature. Again, it was a famine that provided insights. The trail led to China. In the late 1950s, Mao Zedong decreed that all men and women had to work in steel production. Nobody took care of the fields. The wheat rotted for 3 years. In their despair, people started eating bark. 15 million people died, and the incidence of schizophrenia in China doubled in the subsequent generation. This picture fits in with a study from Denmark. Children born during the summer months suffer from schizophrenia a lot less often than winter children, because the food quality is worse in winter.
You need food that's very high in methyl groups. That could be folate, which is contained in aubergines and beans, for example. If I eat these foods, then the enzymes that can deliver methyl remain active. We know about the connection between methylation and gene activity. We can even go so far as to say that through the foods one eats, one can greatly regulate enzyme activity, or which genes are switched on or off.
Our bodies are constantly renewing their cells through cell division. No part of us is older than 7 years. The materials for this lifelong building site come from our food. In order to divide, cells have to accomplish an unimaginable feat. Our DNA contains 3.2 billion base pairs. If this cell division takes place over 20 hours, our cells have to move more than 44,000 base pairs to the right location every second. If cells don't have all the necessary building materials because of malnutrition, errors can creep in. Information is lost. Eating disorders such as bulimia have long ago become part of modern society. But even models who starve themselves skinny want to have children one day. What if all of the building materials cells need for a healthy growth in the womb, such as folic acid and vitamin B12, aren't available because the mother is constantly vomiting during pregnancy?
Very many women vomit in early pregnancy. And we've always thought that this wasn't a big problem because this problem goes away after a few weeks or months. And the baby is often seen as a parasite who just takes whatever it needs. And this study has shown that even undernutrition in early gestation without any later undernutrition has big effects on their health. So we're now actually studying women who suffered from extreme nausea and vomiting in early pregnancy and try to see whether their children are less healthy, and if so, whether we can help these women with extra feeding in early pregnancy.
How life on Earth was able to develop from the interplay of the elements in the first place, how single-celled organisms evolved into the species diversity we have today, and how life can adapt to constantly changing dangers and sudden changes is amongst the greatest mysteries of life. As long as we don't have the answers, we can only look at it as a miracle that needs to be researched. The laws of evolution, with its instruments of mutation and selection, only offer a partial explanation for this miracle. Epigenetics can work on a much faster timescale, switching genes on and off in a single generation. It allows fast responses to environmental changes. Epigenetics has rapidly become the biological boom science. Hence, the scientists of the European Epigenome Network are meeting in Madrid to exchange all the latest research results. The atmosphere is euphoric.
If it shifts, it shows the difference. It's absolutely huge. The reason it's huge is because it's something that people can relate to and must relate to. It's basically how the environment influences our genome. It's how the environment affects the way genes work. It's of fundamental importance to everyone.
And this is a young generation that grew up with a developed sense of awareness of their diets. Their research results provide scientific evidence of why certain foods were held in high regard in the past. Cod liver oil and seafood activate genes in winter that produce antibodies. Certain fruits in summer guard against uncontrolled cell growth, cancer. The old masters immortalized many of the epigenetically most active foods without even knowing it. The first to suspect the power of broccoli was Catherine de' Medici, Queen of France. Catherine's 10 children were extremely weak, and some of them died very young. Then she had broccoli plants brought in from her Italian home country, and henceforth all of her children survived.
Epigenetics goes back to the old question of whether our DNA contains all of the information that determines our development. Is it enough to know our DNA sequence? The current outstanding publications, like the decryption of the human genome and of many other model organizations, showed us that knowing the DNA is not enough. The human DNA sequence contains more than 3 billion letters, more than 25,000 genes, but that information is not enough to say, this cell is a stem cell, This cell is an old cell. This cell is a sick cell. It's not enough.
By the switching of genes on and off, chromosomes can have many different combinations of active genes. This means there is a second level of information, our epigenome, a biochemical trick that manages to achieve incredible things with our compact DNA. A sensational discovery has recently been made in Switzerland, which could have unimagined effects on research. At the university in Basel, epigeneticist Renato Parro has heated up the eggs of special fruit flies to 37 degrees.
These special flies change their eye color from white to red when I raise the temperature. If we expose embryos to heat, the flies are born with red eyes.
But Parro and his colleagues only discovered the actual sensation when they also inspected the eye color of these flies' offspring. Something had happened that wasn't supposed to happen.
We found out that the offspring of these flies also had red eyes, even though they hadn't been exposed to any external environmental factors, such as raised temperatures. It turned out this trait could be passed on epigenetically.
Epigenetic inheritance means that acquired characteristics are passed on to the next generation. This claim was very much frowned upon for a long time, but it has already been proved in plants.
There are several examples in plants where epigenetic characteristics are passed on to subsequent generations. There are color changes in seeds that are passed on, and petal color based on epigenetic changes can be too. I think epigenetics allow fast reactions, and if they are good for the plants, they can be passed on. A large number of epigenetic processes have been described in plants, and we're finding the same mechanisms at work in animals. Genomic imprinting was described in plants in the early 70s, and 10 years later it was found in mice.
Acquired characteristics can thus be passed on, a claim that was considered absurd for a long time. Does this work in humans too?
I think there is pretty much good evidence now that epigenetic inheritance does exist. Especially in response to environmental cues.
The results of the Dutch doctor Tessa Rozeboom in her study of the winter of famine confirms epigenetic inheritance.
If you're born as a baby with the weather forecast of harsh conditions, you are very efficient in processing food. And you have a normal Western diet in our terms, you are so efficient that you're storing a lot of fat, and you have more fat in your arteries and are more likely to get a heart attack, for instance.
She has examined her test subjects for the inheritability of epigenetic changes.
My daughter frequently gets bladder infections. I don't know if that has something to do with the winter of famine.
The doctor wants to see for herself. She wants to meet the daughter, who works on a nearby playground. At first glance, she's a vivacious person. Upon closer questioning by the scientist, there are other complaints besides the bladder weakness.
I like riding, but this year I had this itch. First I thought it was the straw and the hay, but then I found out I have a pollen allergy.
Ms. Fafiani's daughter reports having allergies, and that fits very nicely with the results we've found so far in that babies of children who themselves were exposed to famine while they were in their mother's womb have more diseases, are less healthy in general, and especially more often report things like allergies, and that's exactly what she's reporting.
That would mean that we don't just pass on our genes from generation to generation, but also our epigenetic information, a scary notion. Babies in the womb and newborns sound out the food situation they face. The food given to them in the first months of life programs their bodies for the rest of their lives. It's not just the food that's important, the packaging matters too. Baby bottles made of polycarbonate contain a substance that leaches in small quantities.
Bisphenol A is the fundamental building block for plastics and coatings that are used for products where the main aims are longevity, heat resistance, and safety. These are things like bicycle helmets, safety goggles, CDs, DVDs, windscreens, mobile phones, and household appliances.
It's also in baby bottles, and when they are heated up, molecules leach from the plastic and are consumed by babies along with the food. Bisphenol A isn't toxic, but it's not without its problems.
Germany's Federal Institute of Risk Evaluation has been looking at bisphenol A for a long time because it's an important industrial chemical. We have information that this substance isn't toxic or carcinogenic, but it is capable of producing hormone-like effects.
Bisphenol A likes to dock to our estrogen receptors because of its molecular structure. These receptors then become active and can switch genes on that should be switched off.
There are various scientific publications that indicate that even minute quantities of bisphenol A, if they are ingested, cause changes regarding puberty, for example. They can cause changes to mammary gland growth, to the size of the womb, or to the vagina.
Researchers now suspect bisphenol A can even program a girl's body to go into puberty. You can already see some girls developing breasts, she says. Those girls would like to be children again. I think I'm still too young. I'm only 9. In animal experiments, the connection between sexual maturity and this omnipresent chemical has already been proved. The European authorities still see no reason to act when it comes to humans, even though constant heating leads to severe wear and first teeth do the rest.
One important issue when it comes to evaluating the effect of the substance is that bisphenol A is metabolized fully and very quickly by humans.
The human metabolism thus immediately eliminates this artificial hormone again. But how then can the results obtained at Berlin's Charité hospital be explained?
We were the first working group in the world to be able to prove that the substance bisphenol A can be found in the blood of pregnant women as well as in their newborn babies.
Genes are already being switched on and off in fetuses in order to prepare them for life as well as possible. Some of these switches remain active for several generations. Are we already manipulating the genes of our grandchildren without even knowing it? The artificial hormone bisphenol A doesn't just occur in babies' bottles. The majority of tins used for food are coated with bisphenol A containing substances, and after the tins are filled, they are briefly heated up. A lifelong tendency to obesity could also be the result of this substance, according to researchers, because in animal experiments it was possible to prove this on rats.
There are studies based on animal experiments that indicate that bisphenol A can influence the production of fat cells and thus weight.
Schoenfelder's study, along with 160 other papers, warns about the risks of bisphenol A. Now, plastics companies have commissioned some extremely laborious papers following a specific pattern. In them, bisphenol A is characterized as completely harmless.
All of the studies that were conducted using this design have clearly shown that this substance is completely risk-free.
There is no reason to question bisphenol A in its applications. These products are good and they are safe.
Other countries don't think the matter is this clear-cut.
Canada has published a statement saying that plastics containing bisphenol A should be kept out of areas affecting pregnant women and newborns. I welcome this decision.
Parents who don't want to take any risks are already using plastic products that are bisphenol A free, or else glass bottles, particularly in the United States and Japan. All that's supposed to be in these babies' bottles is their food, and not synthetic estrogen. What we eat influences our health, often for the rest of our lives. That's why the diets of pregnant women and small children are so important. Just how important is only beginning to be understood.
Epigenetics started like a study in cancer, but now we realize that's important for other diseases like mental disorders, like Alzheimer, like cardiovascular disease. And I think that is one of the next frontiers in the research of human diseases.
Our digestive tract is an area often affected by cancerous tumors. In Berlin, epigeneticists have joined forces with doctors so that this young science can be made available to cancer prevention as soon as possible.
It is very advantageous to discover bowel cancer early on. Of course, it's better to discover a problem before it turns cancerous, so the cancer can be prevented. If it's diagnosed early, patients have a very good chance of recovery. Later on, the situation is a lot more serious.
To prevent things from getting serious, a new procedure has been developed in Berlin. A regular blood sample is enough. The patient's DNA can then be extracted from the blood plasma.
Blood plasma contains DNA fragments that come from the tumor. Cells die in the tumor. They give off DNA, and this DNA finds its way into the bloodstream. When we take a blood sample, we can find the tumor's DNA, but only because the DNA methyl fingerprint is different from the fingerprint of the DNA that is in the blood of a healthy patient.
The methylation pattern in cancer patients is the result of the genes that are switched on or off in the tumor. If these patterns are found, bowel cancer is also likely. Although this method is still in the trial period, the advantages of epigenetic diagnosis are evident. Cancer can be detected a lot sooner during routine examinations. Epigenetic medicine could treat diseases before they even break out. The right diet can probably prevent the worst. The superstars of an epi-diet are avocados, broccoli, and pomegranates, whose miracle effects have long been touted. Lettuce, cereals, and green leafy vegetables are further good sources of folic acid, a substance that plays a crucial role in the methylation of genes. Our genes are also happy to get something from Granny's cookbook, like beans and liver. The best advice is to eat a balanced diet, because that's the food for the next generation.
There are indications that some foods are especially important in pregnancy. Fatty fish, for instance, is very important to consume, and green leafy vegetables are important.
We cannot blame our genetics for our behavior of our disease 100%. We have some responsibility here. We can change how these genes are expressed thanks to epigenetics, and this relates in the way we behave and the way we expose to different carcinogens or different substances from the environment.
The issues regarding dietary composition, vitamin metabolism, and other such matters are important for epigenetics.
Yes, of course, we will be able to understand how this works. And understanding how this works, it also means I will be able to prevent it.
Especially cancer patients could benefit from epigenetically efficacious medications.
Since epigenetic changes aren't so set in stone as genetic changes, we can be hopeful that substances could be found to revert these epigenetic changes again.
The old wisdom, you are what you eat, has turned out to be true. We are all switching our genes on and off with our diets. For this reason, we are controlling our appearance, behavior, and health, often for our whole lives, and maybe even for the next generation.